Microbiology Chapter 11, Mechanisms of Microbial Genetics, Part 2. In this section, we will compare different types of mutations, including missense, nonsense, and silent mutations, and describe the differences between light and dark repair mechanisms. We will also learn how various mutagens act and understand how the Ames test can be used to detect carcinogens. Additionally, students will analyze DNA sequences to identify examples of different types of mutations. A mutation is a heritable alteration in an organism's DNA sequence, which can result in a mutant displaying a noticeable change in phenotype compared to the wild type, the most common phenotype found in nature. This DNA change is transcribed into mRNA, potentially altering the amino acid sequence of a protein during translation. Since proteins are responsible for most cellular functions, any alteration in their amino acid sequence can lead to changes in the phenotype of both the cell and the organism. Mutations in DNA can be categorized based on how they modify the DNA sequence. A common type is a point mutation, which involves substituting one base for another. Additionally, mutations can occur through insertions, where one or more bases are added, or deletions, where one or more bases are removed from the DNA sequence. Effect of mutations on protein structure and function. Point mutations and their effect on protein functions first. Point mutation can influence how proteins function in various ways. Due to the redundancy in the genetic code, a point mutation often results in the same amino acid being incorporated into the protein, despite a change in DNA sequence. This type of mutation is referred to as a silent mutation because it does not alter the protein structure. Conversely, a missense mutation occurs when a different amino acid is added to the protein. The effect of this mutation depends on how chemically distinct the amino acid is from the original, as well as its position within the protein. For instance, if the altered amino acid is situated in the enzyme's active site, the missense mutation could have a considerable impact. Many missense mutations lead to proteins that can still function to some degree. Sometimes the effect of missense mutation only manifests under specific environmental conditions. These are known as conditional mutations. In rare cases, a missense mutation may provide selective advantage to the organism that carries it in certain environments. Another type of mutation is a nonsense mutation, which converts a codon that typically codes for an amino acid into a stop codon. This results in a shorter proteins that are generally non-functional compared to the wild type. Effect of deletions and insertions in DNA. Deletions and insertions in DNA can produce varying effects on protein functionality. Codons consist of 3 nucleotides, so when nucleotides are inserted or deleted in groups of 3, these can lead to addition or removal of one or more amino acid without significantly affecting the protein's function. However, frame-shift mutations occur when nucleotides are inserted or deleted in quantities that are not multiples of 3. This results in a shift in the reading frame of the genetic code. Since ribosomes read mRNA in triplet codons, frame-shift mutations can impact every amino acid following the mutation point. Furthermore, this new reading frame might introduce a stop codon before the full coding sequence is translated. As a result, proteins produced from genes with frameshift mutations are typically non-functional. Mistakes during DNA replication can lead to spontaneous mutations. The error rate of DNA polymerase is approximately 1 incorrect base for every billion base pairs that are replicated. Additionally, exposure to mutagens can result in induced mutations, which are various types of chemical agents or radiation, as indicated in the table. Being exposed to a mutagen can increase the mutation rate by more than 1,000 times. Many mutagens are classified as carcinogens, which are agents that can cause cancer. However, it is important to note that while nearly all carcinogens are mutagenic, not all mutagens are necessarily carcinogenic. Various types of chemical mutagens interact directly with DNA by acting as nucleoside analogs or by modifying nucleotide bases. Chemicals known as nucleoside analogs resemble normal nucleotide bases in structure and can be incorporated into DNA during replication. These base analogs induce mutations because they often follow different base pairing rules compared to the bases they replace. Other chemical mutagens can alter normal DNA bases, leading to distinct base pairing rules. For example, nitrous acid deaminates cytosine, transforming it into uracil. In this case, uracil pairs with adenine during the next round of replication, changing a GC base pair into an AT base pair. Additionally, nitrous acid deaminates adenine to hypoxanthine, which pairs with cytosine instead of thymine, resulting in the conversion of a TA base pair into CG base pair. Chemical mutagens known as intercalating agents function in a unique manner. These molecules slide between the stacked nitrogenous bases of the DNA double helix, distorting the structure and creating atypical spacing between nucleotide base pairs. As a result, during DNA replication, DNA polymerase may either skip replicating several nucleotides, leading to a deletion, or insert extra nucleotides, resulting in an insertion. Either outcome can cause a frameshift mutation. Combustion products such as polycyclic aromatic hydrocarbons are especially hazardous intercalating agents that can contribute to mutation-driven cancer. Commonly used for intercalating agents in the laboratories, such as ethidium bromide and acridine orange, serve to stain DNA for visualization and are also potential mutagens. In this slide photograph, a researcher is observing a DNA samples run on an agarose gel. The DNA is stained with ethidium bromide, shows an orange fluorescence under a UV trans illuminator. As ethidium bromide is an intercalating carcinogen, certain laboratory protocols must be practiced to handle it and dispose it. Exposure to the either ionizing or non-ionizing radiation can induce mutations in DNA, but they do so through different mechanisms. Strong ionizing radiation, such as X-rays and gamma rays, can cause both single and double-stranded breaks in the DNA backbone by forming hydroxyl radicals upon radiation exposure. Additionally, ionizing radiation can modify DNA bases. For instance, it can convert cytosine into uracil through a process similar to the action of nitrous acid. This type of radiation is also utilized to kill microbes for sterilizing medical devices and foods due to significant non-specific effect on damaging DNA, proteins, and other cellular components. See using physical methods to control microorganisms that we are going to study in the subsequent sections. In contrast, non-ionizing radiation such as ultraviolet light does not possess enough energy to trigger these chemical alterations. However, it can cause dimer formation between two adjacent pyrimidine bases, typically involving two thymines, within a nucleotide strand. During the formation of thymine dimers, the two adjacent thymine become covalently bonded. If these dimers are not repaired, DNA replication and transcription can be halted at this location. DNA polymerase might attempt to replicate the dimer incorrectly, which could result in frameshift or point mutations. This slide figure shows a photograph of an X-ray technician with radiation shielding. X-ray is an ionizing radiation with more penetrating power and require proper shielding. On the right-hand side, you are seeing an agarose gel on a laboratory UV trans illuminator. The acrylic shielding is enough to block the UV. UV is non-ionizing radiation with much less penetrating power. The process of DNA replication is highly accurate, but mistakes can occur spontaneously or be induced by mutagens. Uncorrected mistakes can lead to serious consequences for the phenotype. Cells have developed several repair mechanisms to minimize the number of mutations that persist. DNA polymerases are the enzyme that build DNA in cells. During replication, most DNA polymerases can check their work with each base that they add. This process is called proofreading. If the polymerase detects a wrong nucleotide has been added, it will remove and replace the nucleotide right away before continuing with DNA synthesis. Many errors are corrected by proofreading, but a few slip through. Mismatch repair happens right after new DNA has been made, and its job is to remove and replace mispaired bases. Mismatch repair can also detect and correct small insertions and deletions that happen when polymerase slips, losing its footing on the template. How does mismatch repair work? First, a protein complex recognizes and binds to the mispaired base. A second complex cuts the DNA near the mismatch, and more enzymes chop out the incorrect nucleotide and surrounding patch of DNA. A DNA polymerase then replaces the missing section with correct nucleotides, and an enzyme called DNA ligase seals the gap. One thing you may wonder is how the proteins involved in DNA repair can tell who is right during mismatch repair. That is, when two bases are mispaired, like G and T in the drawing above, which of the two should be removed and replaced? In bacteria, original and newly made strands of DNA can be told apart by a feature called methylation state. An old DNA strand will have methyl groups attached to some of its bases, while the newly made DNA strand will not yet have gotten its methyl group. So the base from the new strand is removed and not the old. In eukaryotes, the processes that allow the original strand to be identified in mismatch repair involve recognition of nicks, single-stranded breaks, that are found only in newly synthesized DNA. Base excision repair is a mechanism to detect and remove certain types of damaged bases. A group of enzymes called glycosylases play a key role in base excision repair. Each glycosylase detects and removes a specific kind of damaged base. For example, a chemical reaction called deamination can convert a cytosine base into uracil, a base typically found only in RNA. During DNA replication, uracil will pair with adenine rather than guanine, as it would if the base was still cytosine. So an uncorrected cytosine to uracil change can lead to a mutation. To prevent such mutations, a glycosylase from the base excision repair pathway detects and removes the deaminated cytosines. Once the base has been removed, the empty piece of DNA backbone is also removed, and the gap is filled and sealed by other enzymes. Nucleotide excision repair is another pathway used to remove and replace damaged bases. Nucleotide excision repair detects and corrects type of damage that distort the DNA double helix. For instance, this pathway detects bases that have been modified with bulky chemical groups, like the ones that get attached to your DNA when it is exposed to chemicals in cigarette smoke. Nucleotide excision repair is also used to fix some types of damage caused by UV radiation. The most common type of linkage, a thymine dimer, consists of two thymine bases that react with each other and become chemically linked. In nucleotide excision repair, the damaged nucleotides are removed along with the surrounding patch of DNA. In this process, a helicase cranks open the DNA, form a bubble. A DNA cutting enzyme chop out the damaged part of the bubble. A DNA polymerase replaces the missing DNA, and the DNA ligase seals the gap in the backbone of the strand. The overview of this repair were discussed in the previous two slides. The production of thymine dimers is common since many organisms cannot avoid ultraviolet light, leading to the evolution of mechanisms to repair these lesions. In nucleotide excision repair, also known as dark repair, enzymes removed the pyrimidine dimer and replace it with the correct nucleotides. In E. coli, an enzyme complex scans the DNA. If a distortion in the double helix caused by pyrimidine dimer is detected, the enzyme complex cuts the sugar phosphate backbone several bases upstream and downstream of the dimer. The segment of DNA between these two cuts is then enzymatically removed. DNA polymerase I replaces the missing nucleotides with the correct ones, and DNA ligase seals the gap in the sugar phosphate backbone. The direct repair, also called light repair, of thymine dimers occurs through photoreactivation in the presence of a visible light. An enzyme known as photolyase recognizes this distortion in the DNA helix caused by the thymine dimer and binds to it. Then, in the presence of visible light, the photolyase enzyme changes the shape and breaks apart the thymine dimer, allowing the thymines to correctly base pair with the adenines on the complementary strand. Photoreactivation seems to be present in all organisms except for placental mammals, including humans. This process is particularly important for organisms that are chronically exposed to ultraviolet radiation, such as plants, photosynthetic bacteria, algae, and corals, to prevent the accumulation of mutation caused by thymine dimer formation. One common technique used to identify bacterial mutants is called replica plating. This technique is utilized to detect nutritional mutants known as auxotrophs, which have a mutation in a gene that encodes an enzyme involved in the biosynthesis pathway of a specific nutrient, such as an amino acid. Consequently, while wild-type cells can grow normally on a medium that lacks the specific nutrient, auxotrophs are unable to grow on such medium. During replica plating, as illustrated in the figure slide, a population of bacterial cells is mutagenized and then spread as individual cells on a complex, nutritionally complete plate, allowing them to grow into colonies. Cells from these colonies are then transferred from this master plate, often using sterile velvet. This velvet, which contains the cell, is pressed onto plates of various media in the same orientation. At least one of these plates should be nutritionally complete to ensure that the cells are accurately transferred between the plates. The other plates lack specific nutrients, enabling the researcher to identify different auxotrophic mutants that cannot produce specific nutrients. Cells from the corresponding colony on the nutritionally complete plate can be used to recover the mutant for further study. The Ames test, developed by Bruce Ames in the 1970s, is a method that utilizes bacteria for rapid and cost-effective screening of carcinogenic potential of a new chemical compounds. This test measures the mutation rate associated with exposure to the compound. An elevated mutation rate may indicate that exposure to this compound is linked to a higher risk of cancer. The test organism used is a strain of Salmonella typhimurium that is a histidine auxotroph, meaning it cannot synthesize its own histidine amino acid due to a mutation in a crucial gene necessary for its synthesis. After exposure to a potential mutagen, these bacteria are plated on a media that lacks histidine. The number of mutants that regain the ability to synthesize histidine is recorded and compared to the number of such mutants that occur without the potential mutagen. Chemicals that are more mutagenic will result in a higher number of mutants with restored histidine synthesis in the Ames test. Since many chemicals are not directly mutagenic but can be metabolized into mutagenic forms by liver enzymes, rat liver extract is often included at the beginning of the experiment to simulate liver metabolism. Once the Ames test is completed, compounds identified as mutagenic undergo further testing for their potential carcinogenic properties using other models, including animal models such as mice and rats. In this section, we will compare the processes of transformation, transduction, and conjugation, and explain how asexual gene transfer contributes to the genetic diversity of prokaryotes. Additionally, we will understand the structure of transposons and discuss their implications for bacterial genetic diversity. Vertical gene transfer. When discussing genetic transfer, we typically focus on vertical gene transfer, which is the passage of genetic information from one generation to the next and serve as a primary method of genetic transmission in all cells.
In organisms that reproduce sexually, genetic diversity arises from crossing over events and independent assortment of chromosomes during meiosis. Additionally, sexual reproduction combines the genetic information from two parents, each contributing different genetic traits, resulting in a new combination of genotypes in the diploid offspring. Mutations also play a role in enhancing genetic diversity within a population. The genetic variation is particularly advantageous in fluctuating and unpredictable environments, potentially explaining the evolutionary success associated with sexual reproduction. Horizontal gene transfer, or HGT. When prokaryotes and eukaryotes reproduce asexually, they pass on nearly identical copies of their genetic material to their offspring through vertical gene transfer. While asexual reproduction allows for a quicker production of more offspring, it results in a loss of genetic diversity among these offsprings. To create genetic diversity despite a dominant mode of asexual reproduction, prokaryotes utilize horizontal gene transfer, which is the introduction of genetic material from one organism to another within the same generation. HGT enables even distantly related species to share genes, thereby influencing their phenotypes. It is believed that HGT is more common in prokaryotes, although only a small portion of prokaryotic genome may be transferred at any given time. As research into this phenomenon advances, it may be found to occur even more frequently. Many scientists consider HGT and mutation to be the key sources of genetic variation, providing essential material for natural selection in prokaryotes. While HGT tends to happen more often among evolutionarily related organisms, it can occur between two species that coexist in a natural community. HGT in prokaryotes is known to occur by three primary mechanisms: transformation, where naked DNA is taken up from the environment; transduction, genes are transferred between the cells by a virus; conjugation, use of a hollow tube called conjugation pilus to transfer genes between the cells.
Frederick Griffith and discovery of transformation. Frederick Griffith was the first scientist to show the process of transformation. In 1928, he discovered that live non-pathogenic Streptococcus pneumoniae bacteria could be changed into pathogenic bacteria when exposed to a heat-killed pathogenic strain. He proposed that some kind of agent, which he termed the transforming principle, was transferred from the dead pathogenic bacteria to the live non-pathogenic bacteria. Later in 1954, scientists Oswald Avery, Colin MacLeod, and Maclean McCarty confirmed that the transforming principle was DNA. In the process of transformation, a prokaryote, like a bacteria, take up naked DNA found in its environment, which comes from other cells that have lysed and released their contents, including their genetic material. Many bacteria are naturally competent, meaning they can actively bind to environmental DNA, transport it across their cell membrane into their cytoplasm, and convert it into single-stranded DNA. Typically, double-stranded foreign DNA is destroyed by nucleases as a defense mechanism against viral infections. However, these nucleases usually do not affect single-stranded DNA, allowing it the chance to recombine with bacterial genome. When DNA from different organisms combine, it is referred to as a recombinant DNA. If a bacterium incorporates this new DNA into its genome through recombination, it may gain new traits or phenotypic properties. For instance, if a non-pathogenic bacterium takes up DNA that encodes a toxin gene from a pathogen and integrates it into its chromosome, it could become pathogenic. Plasmid DNA can also be taken up by competent bacteria, providing new characteristic to the cell. Transformation in nature is generally an inefficient process due to the low levels of environmental DNA and the activity of nucleases released during the cellular lysis. Additionally, the genetic recombination process is not always effective in integrating new DNA sequences into the genome. In nature, bacterial transformation plays a crucial role in acquiring genetic elements that encode virulence factors and antibiotic resistance. Genes that provide resistance to antimicrobial substances are found widely in nature, including in environments not influenced by human activity. These genes enable microbes in mixed communities to compete for limited resources and can be transferred within a population through transformation, as well as other forms of horizontal gene transfer. In laboratory settings, we can utilize the natural process of bacterial transformation for genetic engineering, allowing for creation of various medicinal products. Transduction in bacteria. Viruses that infect bacteria, known as bacteriophages, can transfer small segments of chromosomal DNA between bacteria through a process called transduction. In generalized transduction, any part of a bacterium's chromosomal DNA can be accidentally packaged into a phage head during the assembly of the virus and then transferred to another host cell. On the other hand, specialized transduction occurs when a lysogenic prophage, a viral DNA integrated into the bacterial chromosome, is imprecisely excised from the bacterial chromosome. This process can result in the transfer of adjacent bacterial DNA along with the prophage to a new host cell. This transfer can lead to the host acquiring new characteristics, a phenomenon known as lysogenic conversion. Medical significance. A lysogenic phage can carry a virulence genes to its new host, which may enable a new host to become pathogenic. Notably, some pathogenic bacteria like Corynebacterium diphtheri, which causes diphtheria, and Clostridium botulinum, which cause botulism, become virulent due to the introduction of toxin-encoding genes through lysogenic bacteriophages. This highlights the importance of transduction in the transfer of genes that contribute to infectious diseases. Additionally, archaea possesses their own viruses that can also move genetic material between individuals. In conjugation, DNA is transferred directly from one prokaryote to another using a structure known as conjugation pilus. This pilus facilitates contact between the two organisms. In E. coli, the genes responsible for the ability to conjugate are found on the bacterial plasmid called the F plasmid, which is also referred to as a fertility factor. The pilus involved in this process is known as the F pilus. The genes present on the F plasmid encode the protein that make up the F pilus, as well as those that participate in rolling circle replication of the plasmid. Cells that contain the F plasmid and can produce an F pilus are termed as F plus or donor cells, while those that do not possess the F plasmid are classified as F minus or recipient cells. Conjugation of the F plasmid. During the process of conjugation in E. coli, the F pilus of an F plus cell touches an F minus cell and retracts, bringing the cell membranes together. This leads to a formation of a cytoplasmic bridge at the location of the conjugation pilus. As the F plasmid undergoes rolling circle replication in the F plus cell, a single-stranded copy of the F plasmid is sent through the cytoplasmic bridge to the F minus cell, which then creates the complementary strand, resulting in a double-stranded plasmid. The F minus cells is now transformed into an F plus cell, which can produce its own conjugation pilus. Over time, in a mixed population of both F plus and F minus cells, all cells will eventually become F plus cells. Additionally, genes on the E. coli F plasmid encode proteins that inhibit conjugation between the F plus cells. In E. coli, standard conjugation typically resulted in transfer of only the F plasmid DNA. However, conjugation can also transfer chromosomal DNA. This occurs between the F plasmid, sometimes integrates into the bacterial chromosome through a process called recombination, forming what is known as an HFR cell. The term HFR stands for high frequency of recombination, which is observed when recipient F- cells receive genetic material from HFR cells during conjugation. Similar to the imprecise excision of a prophage in a specialized transduction, the integrated F plasmid can be imprecisely excised from the chromosome, leading to the formation of an F prime plasmid. This F prime plasmid carries some chromosomal DNA that is adjacent to the integration site. When conjugation occurs, this chromosomal DNA is introduced into the recipient cell, where it may either be retained as a part of the F prime plasmid or recombined into the recipient cell's bacterial chromosome. HFR cells can treat the bacterial chromosome similarly to a large F plasmid, making an effort to transfer a copy of it to a recipient F- cell. Due to the size of the bacterial chromosome, transferring the entire chromosome is a lengthy process. However, since the contact between the bacterial cells during conjugation is brief, it is uncommon for the entire chromosome to be successfully transferred. The chromosomal DNA from the host that is located near where the F plasmid integrates is more likely to be displaced and transferred during the unidirectional rolling circle replication process. This DNA is more likely to recombine into the recipient cell's chromosome compared to genes that are farther away from the integration site. Consequently, the positioning of bacterial genes within the HFR cell's genome can be mapped based on the order in which they are transferred during conjugation. Therefore, before widespread use of bacterial genome sequencing, distances on prokaryotic genome maps were typically measured in minutes. Plasmids are significant elements of extrachromosomal DNA found in bacteria and are considered part of the bacterial genome in those cells that contain them. From a clinical standpoint, plasmids often carry genes that contribute to virulence. For instance, our plasmids include genes that enable bacterial cells to resist specific antibiotics. In addition to antimicrobial resistant genes, our plasmids also possess genes that manage conjugation and the transfer of the plasmid. These plasmids can transfer between the cells of the same species as well as between different species. Typically, a single R plasmid contains multiple genes that can confer resistance to various antibiotics. Moreover, plasmids can encode genes essential for producing different toxins and molecules that aid in the colonization during infections. A notable example is a virotoxin-produced strains of E. coli or VTEC, which appear to have acquired the genes for Shiga toxin from a gram-negative bacterium Shigella dysentery via a large plasmid that carries this toxin. VTEC is known to cause severe diarrheal diseases which may lead to hemolytic uremic syndrome, potentially resulting in kidney failure and death. In non-clinical context, bacterial genes that code for metabolic enzyme necessary to break down specialized atypical compounds, such as polycyclic aromatic hydrocarbons, are often found on plasmids as well. Furthermore, some plasmids can migrate from bacterial cells to other cell types, including those in plant and animals, through mechanisms different from conjugation. These mechanisms and their application in genetic engineering are discussed in detail in the modern applications of microbial genetics. Transposons, the jumping genes. Transposons, also known as a transposable element or jumping genes, are pieces of DNA that have unique sequences called inverted repeats at their ends, along with a gene that encodes the enzyme transposase. These elements allow a DNA sequence to cut itself out from one spot in DNA molecule and insert itself into another location through a process called transposition. The discovery of transposons dates back to the 1940s when American geneticist Barbara McClintock found them in maize. Since then, transposons have been identified in all types of organisms, including prokaryotes and eukaryotes, which means transposition occurs across various life forms and is not limited to prokaryotes. Most transposons operate in a cut and paste manner, meaning they move from one place to the DNA to another without leaving a copy behind. However, some transposons can also become copy and paste, where they create duplicate of themselves in a new location while retaining their original position. Transposons contribute to genetic diversity by moving within the same DNA molecule or between different molecules and even across cells. This movement can affect an organism's traits by either turning genes off or on. In addition to their own sequences, transposons can carry extra genes with them as they move. For instance, bacterial transposons can relocate antibiotic-resistant genes, shifting them from chromosomes to plasmid. This process is responsible for clustering multiple antibiotic-resistant genes on single R plasmid in Shigella strains, which causes bacterial dysentery. Such R plasmid can then be easily shared with bacterial populations through a method called conjugation that we discussed before. This slide shows a summary of mechanisms that lead to the prokaryotic diversity. In this section, we are going to discuss the regulation of gene expression in bacteria, compare inducible operons and repressible operons, and describe why regulation of operon is important. DNA in multicellular organisms and bacterial cultures. In multicellular organisms, each nucleated cell contains copies of the same DNA. Similarly, in two pure bacterial cultures that originate from the same colony, the DNA is identical, except for variations that occur due to spontaneous mutations. Given that all cells in a multicellular organism share the same DNA, how do cells in different parts of the body display distinct characteristics? Furthermore, how can bacterial cells in two pure cultures, when subjected to different environmental conditions, show different traits? The answer lies in the gene expression. Although the cells are genetically identical, they do not activate or express the same set of genes at any given time. Only a specific subset of proteins is expressed in each cell. Gene structure and regulation. Genomic DNA consists of structural genes, which produce cellular structures or enzymes, and regulatory genes, which control gene expression. The process of gene expression is intricately regulated. In multicellular organisms, regulating gene expression facilitates cellular differentiation, allowing cells to take on unique roles. In a single-celled organism such as prokaryotes, it primarily ensures that the cell conserves resources by avoiding the production of unnecessary proteins. Importance of understanding gene expression. Understanding the mechanism that controls gene expression is crucial for comprehending human health. Malfunction in this process can lead to diseases, including cancer. Additionally, understanding how the gene expression of a pathogen interacts with that of its human host is vital for studying infectious diseases. Gene regulation involves a complex network of interaction within a cell, influenced by environmental signals, internal signaling molecules, and the cell's DNA. These interactions determine which genes are expressed and which are suppressed depending on the circumstances. Comparing prokaryotes and eukaryotes. Both prokaryotes and eukaryotes have similarities in their gene regulation mechanism. However, gene expression in eukaryotes is more complex due to the separation of transcription and translation processes in time and space. While most gene regulations in prokaryotes occur at the transcriptional level, eukaryotic gene regulation involves both transcriptional control and post-transcriptional mechanism, which takes place after the primary transcript has been formed. In bacteria and archaea, related structural proteins are typically encoded together in the genome within a block known as an operon. These genes are transcribed together under a single promoter, resulting in a polycistronic transcript. This organization allows for the simultaneous regulation of all structural genes that encode the enzymes needed for specific biochemical pathways. These pathways often require all enzymes at once or none at all. For instance, E. coli, the genes that encode the enzyme necessary for using lactose as an energy source, are located in the lactose or lac operon. controlled by the lac promoter. The first demonstrate this gene organization in bacteria were French scientists Francois Jacob and Jacques Monod and the Pasteur Institute through their studies of the lac operon in E. coli. They were awarded the Nobel Prize in Physiology or Medicine in 1965 for their contributions. Although eukaryotic genes are not organized into operons, prokaryotic operons serve as an excellent model for understanding gene regulation. Some gene clusters in eukaryote function similar to operons, allowing us to apply many principles of operon function to eukaryotic systems, which helps explain changes in gene expression that can lead to pathological conditions like cancer. Each operon contains DNA sequences that influence its transcription, found in a region called the regulatory region. This region includes a promoter in its surrounding area where the transcription factors proteins encoded by regulatory genes can bind. These transcription factors play a crucial role in the binding of RNA polymerase to the promoter and facilitate its progress in transcribing the structural genes. A repressor is a type of transcription factor that inhibits gene transcription in response to external stimuli by binding to DNA sequence known as the operator. This operator is located between the RNA polymerase binding site, the promoter, and the transcriptional start site of the first structural gene. By binding to the operator, the repressor physically obstructs RNA polymerase from transcribing the structural genes. In contrast, an activator is a transcription factor that promotes gene transcription by enhancing the binding of RNA polymerase to the promoter. Additionally, an inducer is a small regulatory molecule that can either activate or repress transcription by interacting with a repressor or an activator. In prokaryotes, some operons consistently required, exhibiting constitutive expression. This means they are continuously transcribed and translated to maintain constant level of protein products essential for cellular maintenance, including functions like DNA replication, repair, expression, as well as core metabolic processes. On the other hand, other prokaryotic operons are expressed only when necessary and are regulated by repressor, activators, and inducers. Prokaryotic operons are often regulated by binding of repressor proteins to a specific region called the operators, which prevent the transcription of the structural genes. These operons can be grouped into two main types: repressible operons and inducible operons. Repressible operons, such as tryptophan or TRIP operon, usually include genes that encode for enzymes needed in a biosynthetic pathway. When the cell needs the product by this pathway, like tryptophan, the operon remains active. However, once the product accumulates and the cell no longer requires more, the operon is turned off or repressed. Inducible operons, on the other hand, exemplifies by the lac operon found in E. coli, contain genes that produce enzymes for metabolizing specific substrates like lactose. The expression of these operons is only triggered when the substrate is present, meaning the enzymes are produced only when needed. Operons are clusters of genes under the control of a single promoter, allowing for coordinated expression in prokaryotic organisms. The regulation of operons can be classified into two main categories: positive and negative regulation, both of which play crucial roles in cellular metabolism and gene expression. Negative regulation involves the binding of a repressor protein to the operator region of an operon, inhibiting transcription. This prevents the RNA polymerase from binding to the promoter, thus blocking gene expression. An example of negative regulation is the lac operon in E. coli, which is responsible for lactose metabolism. When lactose is absent, the repressor protein binds to the operator, preventing transcription. However, when lactose is present, it binds to the repressor, causing a conformational change that releases it from the operator. This allows transcription of genes required for lactose utilization to occur. Positive regulation, on the other hand, involves the activation of transcription by an activator protein. In this case, the activator binds to a specific site on the DNA, enhancing the binding of RNA polymerase to the promoter and promoting gene expression, which we will discuss later. Bacteria such as an E. coli need amino acids to survive, because like us, they need to build proteins. One of the amino acids they need is tryptophan. If tryptophan is available in the environment, E. coli will take it up and use it to build proteins. However, E. coli can also make their own tryptophan using enzymes that are encoded by 5 genes. These 5 genes are located next to each other in what it is called a trip operon. If the tryptophan is present in the environment, then E. coli don't need to synthesize it, so transcription of these genes in the trip operon is switched off. When tryptophan availability is low, on the other hand, the operon is switched on, the genes are transcribed, biosynthetic enzymes are made, and more tryptophan is produced. Structure of the trip operon. The trip operon includes 5 genes that encode enzymes needed for tryptophan biosynthesis, along with a promoter and an operator. The genes of the trip operon are transcribed as a single polycistronic mRNA. The 5 genes that make up the trip operon encodes the following enzymes of the tryptophan biosynthetic pathway: TRPE, anthranilate synthetase component 1, TRPD, anthranilate synthase component 2, TRPC, N5' phosphoribosyl anthranilate isomerase, or indole 3-glycerol phosphate synthase, TRPB, beta subunit of the tryptophan synthase, TRPA, alpha subunit of the tryptophan synthase. Turning the operon on and off. What does the operator do? This stretch of DNA is recognized by a regulatory protein known as the trip repressor. When the repressor binds to the DNA of the operator, it keeps the operon from being transcribed by physically getting in the way of RNA polymerase, the transcription enzyme. The trip repressor does not always bind to DNA. Instead, it binds and blocks transcription only when tryptophan is present. When tryptophan is around, it attaches to the repressor molecule and changes their shape so they become active. A small molecule like tryptophan switches a repressor into its active state is called a co-repressor. When there is little tryptophan in the cell, on the other hand, the trip repressor is inactive because no tryptophan is available to bind to it and activate it. It does not attach to the DNA or block transcription, and this allows the trip operon to be transcribed by RNA polymerase. In this system, the trip repressor acts as both a sensor and a switch. It senses whether tryptophan is already present at high levels, and if so, it switches the operon to the off position, preventing unnecessary biosynthetic enzymes from being made. More trip operon regulation, attenuation. Like regulation by the trip repressor, attenuation is a mechanism for reducing expression of the trip operon when levels of tryptophan are high. However, rather than blocking initiation of transcription, attenuation prevents completion of transcription. When levels of tryptophan are high, attenuation causes RNA polymerase to stop prematurely when it's transcribing the trip operon. Only a short, stubby leader mRNA is made, one that does not encode any tryptophan biosynthesis enzyme. Attenuation works through a mechanism that depends on the coupling of translation of an mRNA that is still in the process of being transcribed, discussed later in the chapter. Lactose, it's what's for dinner. While that may not sound delicious to us, lactose is the main sugar in milk and you probably don't want to eat it plain. Lactose can be an excellent meal for E. coli. However, they will only gobble up lactose when other better sugars like glucose are unavailable. With that for context, what exactly is the lac operon? The lac operon is an operon or group of genes with a single promoter transcribed as a single mRNA. The genes in the operon encode proteins that allow the bacteria to use lactose as an energy source. The illustration shows a schematic of the E. coli. Lactose enters in the periplasmic space via porins present on the outer LPS membrane layer. From the periplasmic space, lactose is imported into the cell by lactose permease embedded in the cell membrane. Inside the cytoplasm, the following reactions occur. Lactose is converted directly to galactose and glucose in the presence of a catalyst, beta-galactosidase. Lactose also reacts with beta-galactosidase in a side reaction converting lactose to allolactose. Allolactose in the presence of beta-galactosidase forms galactose and glucose. The conversions are shown in the molecular structures. Environment nutrient availability changes gene expression, but how much? In absence of lactose, approximately 3 beta-galactosidase molecules are produced, not 0, in one generation of E. coli. But in presence of lactose, 3,000 beta-galactosidase molecules are produced, a 1,000-fold increase in gene expression. What makes the lac operon turn on? E. coli can break down lactose, but it's not their favorite fuel. If glucose is around, they would much rather use that. Glucose requires fewer steps and less energy to break down than lactose. However, if lactose is the only sugar available, the E. coli will go right ahead and use it as an energy source. To use lactose, the bacteria must express the lac operon genes, which encode key enzymes for lactose uptake and metabolism. To be as efficient as possible, E. coli should express the lac operon only when two conditions are met. Lactose is available and glucose is not available. How are levels of lactose and glucose detected? And how do changes in levels affect lac operon transcription? Two regulatory proteins are involved. One, the lac repressor, acts as a lactose sensor. The other, catabolite activator protein or CAP, acts as a glucose sensor. These proteins bind to the DNA of the lac operon and regulate its transcription based on lactose and glucose levels. Let's take a look at how it works. Structure of the lac operon. The lac operon contains 3 genes, LAC-Z, LAC-Y, and LAC-A. These genes are transcribed as a single mRNA under the control of one promoter. Genes in the lac operon specify proteins that help the cell utilize lactose. LAC-Z encodes an enzyme that splits lactose into monosaccharides, single unit sugars, that can be fed into glycolysis. Similarly, LAC-Y encodes a membrane-embedded transporter that helps bring lactose into the cell. In addition to the 3 genes, the lac operon also contains a number of regulatory DNA sequences. These are regions of DNA to which particular regulatory proteins can bind, controlling transcription of the operon. The following proteins and enzymes are encoded by the lac operon. LAC-Z, beta-galactosidase. This enzyme breaks down lactose into glucose and galactose, which can be readily used by the cell. LAC-Y, lactose permease, a transmembrane protein that transports lactose into the bacterial cell. And LAC-A, transacetylase. The exact function of this protein is not fully understood, but it is thought to be involved in modifying lactose molecules. In this slide figure, the promoter is binding site of the RNA polymerase, the enzyme that performs the transcription. The operator overlaps with the promoter, and when the lac repressor is bound, the RNA polymerase cannot bind to the promoter and start transcription. The cap binding site is a positive regulatory site that is bound by the catabolic activator protein or CAP. When CAP is bound to this site, it promotes the transcription, helping RNA polymerase to bind to the promoter. Let's take a closer look at the lac repressor and CAP and their roles in regulation of the lac operon. The lac repressor is a protein synthesized by a gene LAC-I constitutively. The lac repressor represses transcription of the lac operon. It does this by binding to the operator, which partially overlaps with the promoter. When bound, the lac repressor gets in RNA polymerase's way and keeps it from transcribing the operon. When lactose is not available, the lac repressor binds tightly to the operator, preventing transcription by RNA polymerase. However, when lactose is present, the lac repressor loses its ability to bind DNA. It floats off the operator, clearing the way RNA polymerase to transcribe the operon. The change in the lac repressor is caused by a small molecule, allolactose, an isomer of lactose. When lactose is available, some molecules will be converted to allolactose inside the cell. Allolactose binds to the lac repressor and makes it change shape so it can no longer bind DNA. Allolactose is an example of an inducer, a small molecule that triggers expression of a gene or operon. The lac operon is considered an inducible operon because it is usually turned off or repressed, but can be turned on in the presence of the inducer allolactose. Bacteria have the ability to use different substances as a source of carbon. However, they usually prefer glucose over other options. To manage these, bacteria have developed methods to use alternative substances when glucose is no longer available. They also regulate the expression of genes that produce enzymes for using these alternative sources, ensuring that are activated only when the specific substrate is present. In the 1940s, Jack Monod conducted the pioneering research on the growth of E. coli in the presence of two different substrates at the same time. His studies revealed how bacteria show preference for certain substrate, leading to the creation of dioxic growth curves. In these curves, E. coli initially grows rapidly using the glucose, while the enzyme is breaking down, lactose is not produced. Once the glucose is depleted, the growth rate decreases, triggering the production of an enzyme required for the lactose metabolism. As a result, the growth rate of lactose is slower, which is shown by the less steep growth curve compared to when glucose is available. The illustration shows a cell membrane embedded with 2C glucose permease and a phosphotransferase system or a PTS system. The ability to switch from glucose use to another substrate like lactose is a consequence of the activity of an enzyme called enzyme 2A, E2A. When glucose entering the cell, it is phosphorylated by the enzyme E2B. The glucose 6-phosphate is the first substrate of glycolysis in cellular metabolism. At the end of glycolysis, the pyruvate is produced from phosphoenol pyruvate, and the phosphate is transferred back to E2B via E1, HBr, and E2A, known as phosphotransferase system. When glucose levels drop, cells produce much less ATP from catabolism, and E2A becomes phosphorylated but can't transfer the phosphate to glucose in absence of glucose. In this context, the phosphorylated E2A activates adenylyl cyclase, an enzyme that converts some of the remaining ATP to cyclic AMP, a cyclic derivative of AMP and an important signaling molecule involved in glucose and energy metabolism in E. coli. As a result, CAMP begins to rise in the cell. Depletion of cellular ATP occurs in absence of glucose. No glucose, no energy. At this time, if adenylyl cyclase is activated, it breaks down the remaining ATPs and produces a cyclic form of AMP, adenosine monophosphate, where the 5' carbon and the 3' carbon are bonded by the phosphate group. This cyclic structure, known as cyclic AMP or CAMP in short, will eventually bind to the glucose sensor molecule, catabolite activator protein. When lactose is present, the lac repressor loses its DNA binding ability. This clears the way for RNA polymerase to bind to the promoter and transcribe the lac operon. That sounds like the end of the story, right? Well, not quite. As it turns out, RNA polymerase alone does not bind very well to the lac operon promoter. It might make a few transcripts, but it won't do much more unless it gets extra help from catabolic activator protein or CAP. CAP binds to a region of DNA just before the lac operon promoter and helps the RNA polymerase to attach to the promoter, driving a high level of transcription. CAP isn't always active or able to bind DNA. Instead, it's regulated by a small molecule called the cyclic AMP, shown in the previous slide. Cyclic AMP is the hunger signal made by the E. coli when glucose levels are low. The CAMP binds to CAP, changing its shape and making it able to bind DNA and promote transcription. Without the cyclic AMP, the CAP cannot bind to the DNA and it is inactive. CAP is only active when glucose levels are low, cyclic AMP levels are high. Thus, the LAC operon can only be transcribed at high levels when glucose is absent. This strategy ensures that bacteria only turn on the LAC operon and starts using lactose after they have used up all the preferred energy source or glucose. So when does the LAC operon really turn on? The LAC operon will be expressed at high levels if two conditions are met. Glucose must be unavailable. When glucose is unavailable, cyclic AMP binds to CAP, making CAP able to bind DNA. Bound CAP helps RNA polymerase attach to the LAC operon promoter. And condition 2, lactose must be available. If lactose is available, the LAC repressor will be released from the operator by binding to allolactose. This allows RNA polymerase to move forward on the DNA and transcribe the operon. These two events in combination, the binding of activator and release of the repressor, allow RNA polymerase to bind strongly to the promoter and give it a clear path for transcription. They lead to a strong transcription of the LAC operon and production of the enzyme needed for lactose utilization. Now that we have seen all the moving parts of the LAC operon, let's put what we have learned together to see how the operon reacts to a variety of different conditions, presence or absence of glucose and lactose. First, glucose present, lactose absent. No transcription of the LAC operon occurs. That's because the LAC repressor remains bound to the operator and prevents transcription by RNA polymerase. Also, cyclic AMP levels are low because glucose levels are high, so CAP is inactive and cannot bind DNA. Glucose present, lactose present. Low-level transcription of LAC operon occurs. The LAC repressor is released from the operator because the inducer, allolactose, is present. Cyclic AMP levels, however, are low because glucose is present. Thus, CAP remains inactive and cannot bind to the DNA, so transcription only occurs at low leaky level. The term leaky expression can be compared to a leaky faucet of gene expression. It is not tightly closed and leaking dropwise, not much but very little. Glucose absent, lactose absent. No transcription of LAC operon occurs. Cyclic AMP are high because glucose levels are low, so CAP is active and will bound to the DNA. However, the LAC repressor will also be bound to the operator due to the absence of allolactose, acting as a roadblock to RNA polymerase and preventing transcription. Glucose absent, lactose present. Strong transcription of LAC operon occurs. The LAC repressor is released from the operator because the inducer allolactose is present. Cyclic AMP levels are high because glucose is absent, so CAP is active and bound to the DNA. CAP helps RNA polymerase bind to the promoter, permitting high levels of transcription. This slide shows a comprehensive summary of LAC operon regulation under all the conditions mentioned the slide before. Global response of prokaryotes. In prokaryotes, there are also several higher levels of gene regulation that have the ability to control the transcription of many related operons simultaneously in response to an environmental signal. A group of operons all controlled simultaneously is called a regulon. Alarmons. When sensing impending stress, prokaryotes alter expression of a wide variety of operons to respond in coordination. They do this through the production of alarmons, which are small intracellular nucleotide derivatives. The small nucleotide PPGPP act as global regulator of gene expression in bacteria. The alarmon causes a redirection of transcription so that genes important for starvation, survival, and virulence are favored at the expense of those required for growth and proliferation. On encountering host defense mechanism and other harsh conditions during infection, many operons encoding virulence genes are upregulated in response to alarmon signaling. Knowledge of these responses is key to being able to fully understand the infection process of many pathogens and to the development of therapies to counter this process. Alternate sigma factors. since the sigma subunit of bacterial RNA polymerase confers specificity as to which promoters should be transcribed, altering the sigma factors used is another way for bacteria to quickly and globally change what regulons are transcribed at a given time. The sigma factor recognizes sequences within a bacterial promoter, so different sigma factors will each recognize slightly different promoter sequences. In this way, when the cell senses specific environmental conditions, it may respond by changing which sigma factor it expresses, degrading the old one and producing a new one to transcribe the operons encoding genes which products will be useful under the new environmental conditions. For example, in sporulating bacteria of the genera Bacillus and Clostridium, which include many pathogens, a group of sigma factors control the expression of many genes needed for the sporulation in response to sporulation stimulating signals. Most gene expression in prokaryotes is primarily regulated during the initiation of transcription. However, there are also mechanisms that control both the completion of transcription and translation process at the same time. Since their discovery, these mechanisms have been shown to influence the transcription and translation of various prokaryotic operons. These regulatory mechanisms are unique to prokaryotes because they connect transcription and translation directly, unlike in eukaryotes where these processes occur separately. The process of attenuation involves the presence of a stop signal that indicates premature termination. The stop signal, referred to as the attenuator, prevents the proper function of the ribosomal complex, stopping the process. The attenuator is transcribed from the appropriate DNA sequence, and its effects are dependent on the metabolic environment. In times of need, the attenuator within the mRNA sequence will be bypassed by the ribosome and proper translation will occur. However, if there is not a need for a mRNA molecule to be translated, but the process was simultaneously initiated, the attenuator will prevent further transcription and cause a premature termination. Hence, attenuators can function in either transcription attenuation or translation attenuation. Attenuation of TRP operon. Attenuation is a second mechanism of negative feedback in the TRP operon. While the TRPR repressor decreases transcription by a factor of 70, attenuation can further decrease it by a factor of 10, thus allowing accumulated repression of about 700-fold. Attenuation is made possible by the fact that in prokaryotes which have no nucleus, the ribosome begins translating the mRNA while RNA polymerase is still transcribing the DNA sequence. This allows the process of translation to directly affect transcription of the operon. When there is a high level of tryptophan in the region, it is inefficient for the bacterium to synthesize more. When the RNA polymerase binds and transcribes the TRP gene, the ribosome will start translating. This differs from eukaryotic cell where the RNA must exit the nucleus before translation starts. The attenuator sequence, which is located between the mRNA leader sequence 5' UTR and the TRP operon gene sequence, contains 4 domains 1, 2, 3, and 4, where domain 3 can pair with either domain 2 or domain 4. The attenuator sequence at domain 1 contains instruction for peptide synthesis and requires a tryptophan. A high level of tryptophan will permit the ribosome to translate. The attenuator sequence at domain 1 contains instruction for peptide synthesis that requires tryptophan. A high level of tryptophan will permit ribosomes to translate the attenuator sequence domains 1 and 2, allowing domains 3 and 4 to form a hairpin structure, which results in termination of transcription of the TRP operon. Since the protein coding genes are not transcribed due to the row independent termination, no tryptophan is synthesized. In contrast, a low-level tryptophan means that the ribosome will stall at domain 1, causing the domains 2 and 3 form a hairpin structure that does not signal the termination of transcription. Therefore, the rest of the operon will be transcribed and translated so that tryptophan can be produced. Thus, domain 4 is an attenuator. Domain 4 translation can continue regardless of the level of the tryptophan. The attenuator sequence has its codon translated into a leader peptide, but is not a part of the trip operon gene sequence. The attenuator allows more time for the attenuator sequence domains to form a loop structure, but does not produce a protein that is used in later tryptophan synthesis. At the beginning of the transcribed genes of the trip operon is a sequence of 140 nucleotide termed as the leader transcript or TRIP-L. This transcript includes 4 short sequences designated 1, 2, 3, and 4. 1 is partially complementary to sequence 2, which is partially complementary to sequence 3, which is partially complementary to sequence 4. Thus, 3 distinct secondary structure hairpins can form 1, 2, 2, 3, or 3, 4. The hybridization of strands of 1 and 2 to form 1, 2 structure prevents the formation of 2, 3 structure, while the formation of 2, 3 prevents the formation of 3, 4. The 3, 4 structure is the transcription termination sequence. Once it forms, RNA polymerase will dissociate from the DNA and the transcription of the structural genes of the operon will not occur. Part of the leader sequence code for a short polypeptide of 14 amino acids termed as a leader peptide. This peptide contains 2 adjacent tryptophan residues, which is unusual since tryptophan is fairly uncommon amino acids. About 1 in 100 residues in a typical E. coli protein is tryptophan. If the ribosome attempts to translate this peptide while tryptophan levels in the cells are low, it will stall at either of the 2 trip codons waiting for a tryptophan tRNA. This happens because there are not many tryptophan tRNAs in a cell, as amino acid is rare in proteins. While it is stalled, the ribosome will physically shield 1 from the transcript, thus preventing forming 1, 2 secondary structure. The sequence 2 is then free to hybridize with the 3 sequence, forming a 2, 3 structure, which then prevents the formation of the 3, 4 termination hairpin. RNA polymerase is free to continue transcribing the entire operon. If tryptophan levels in the cells are high, the ribosome will translate the entire leader peptide without interruption and will only stall during translation termination at the stop codon. At this point, the ribosome physically shields both sequences 1 and 2, and sequences 3 and 4 are thus free to form the 3, 4 structure, which terminates transcription. The result is that the operon will be transcribed only when tryptophan is unavailable for the ribosome, while the TRIP-L transcript is constitutively expressed. To ensure that ribosome binds and begins translation of the leader transcript immediately following its synthesis, a pause site exists in the TRIP-L sequence. Upon reaching this site, RNA polymerase pauses transcription and apparently waits for translation to begin. This mechanism allows for synchronization of transcription and translation, a key element in attenuation. A similar attenuation mechanism regulates the synthesis of histidine, phenylalanine, and threonine. Genetic regulation by RNA is widespread in bacteria. One common form of riboregulation in bacteria is the use of ribonucleic acid sequences encoded within mRNA that directly affect expression, transcription, and translation both of genes encoded in the full transcript, called cis-acting element because they act on the same molecule they are coded in. These regulatory elements are known as riboswitches and are defined as mRNA elements that bind metabolites or metal ions as ligands and regulate mRNA expression by forming alternative structure in response to this ligand binding. To date, scientists have discovered that a variety of ligands are sensed by riboswitches. The group includes magnesium ion, nucleic acid precursor, enzyme cofactors, and amino acid residues. Where are riboswitches located? Ribose switches are often located at the 5' untranslated region or 5' UTR, a stretch of the RNA that precedes the translation start site of the bacterial mRNA. The mechanism. The function of the ribose switches is tied to the ability of the RNA to form a diversity of structure. The most basic of this double-stranded helix similar to that found in DNA. However, since most RNAs, unlike DNA, do not need to maintain perfect Watson-Crick base pairing, they can form other types of structure. For example, a single-stranded RNA molecule can fold back on itself to form a hairpin, which is composed of a helix capped by a loop. These elements of structures are called the secondary structures. Ribose switches are composed of two domains, the aptamer domain, which is the pink in the illustration, and the expression platform, which is purple or brown in the illustration. The aptamer domain acts as a receptor that specifically binds to a metabolite. The expression platform acts directly on the gene expression through its ability to toggle between two different secondary structures in response to ligand bounding. Common to both domains is something called switching sequence, and its placement in the aptamer domain or the expression platform ultimately dictates the expression outcome of the mRNA. Specifically, if metabolite binding to the ribose switch stabilizes the incorporation of switching sequence into the aptamer domain, the expression platform must fold into specific structures. Figure A shows the ribose switches that control the transcriptional repression, having a switch sequence that directs the formation of a row-independent transcription terminator, a short stem loop-like structure followed by 6 or more uridine residues. That signals RNA polymerase to abort transcription. In figure B, the ribose switch that regulate a translational initiation utilize a switching sequence that can expose or occlude ribosome binding site, called a Shine-Dalgarno sequence. Although the focus on our discussion of transcriptional control use prokaryotic operons as examples, eukaryotic transcriptional control is similar in many ways. As in prokaryotes, eukaryotic transcription can be controlled through the binding of transcription factor, including repressor and activators. Interestingly, eukaryotic transcription can be influenced by binding of proteins to regions of DNA called enhancer, rather far away from the gene, through DNA looping facilitated between the enhancer and the promoter. Overall, regulating transcription is a highly effective way to control gene expression in both prokaryotes and eukaryotes. However, the control of gene expression in eukaryote in response to environmental and cellular stresses can be accomplished in additional ways without the binding of transcription factors to regulatory regions. In eukaryotes, the DNA molecules or associated histones can be chemically modified in such a way as to influence transcription. This is called epigenetic regulation. Methylation of certain cytosine nucleotides in DNA in response to environmental factors has been shown to influence use of such DNA for transcription, with DNA methylation commonly correlating to lower level of gene expression. Additionally, in response to environmental factors, histone proteins for packaging DNA can also be chemically modified in multiple ways, including acetylation and deacetylation, influencing the packaging state of the DNA and thus affecting the ability of loosely wound DNA for transcription. These chemical modifications can sometimes be maintained through multiple rounds of cell division, making it at least some of these epigenetic changes heritable. Clinical focus: necrotizing fasciitis. Mark, a 60-year-old software engineer with a type 2 diabetes, was managing his condition through diet and exercise. One spring morning, while gardening, he scraped his lower leg on blackberry brambles. He ignored the wound for the rest of the day and didn't clean and treat it until later in the evening. Over the next 2 days, his leg became red, swollen, warm, and sore. He developed a fever and stiffness, prompting him to seek medical attention when he felt increasingly weak. In the emergency department, a nurse confirmed that Mark had made the right choice to come in. His symptoms indicated an out-of-control infection. The affected area showed increased swelling or rash, blistering, and gray skin, with a foul smell from pus, and the physician diagnosed him with necrotizing fasciitis and ordered a culture of the blister fluid and blood work. Mark was admitted to the intensive care unit for intravenous broad-spectrum antibiotics. Despite treatment, his conditions worsened with confusion, dizziness, low blood pressure, rapid breathing, and intensified blistering. Possible causes of Mark's necrotizing fasciitis include strains of bacteria that can lead to severe infection, such as Group A Streptococcus. Mark's infection continued to spread, necessitating ventilation support and surgical removal of infected tissue. Despite monitoring and multiple surgeries, the infection was slow to contain. Eventually, Mark was taken off the ventilator, but had lost significant skin and soft tissue. Removing infected tissue helps stem the infection by eliminating the source of bacteria. However, complications may include prolonged recovery, the need for further surgeries, and potential mobility issues. Mark survived, but required skin grafting and physical therapy. He was unlikely to regain full strength in his leg. Laboratory tests confirmed his infection was caused by a Group A Streptococcus, which was reported to health authorities for further analysis. Researchers found that this strain exhibits methicillin resistance, complicating treatment due to limited blood flow in the infected area. Bacterial toxins hindered the effectiveness of antibiotics, making intravenous therapy necessary to prevent sepsis, a common consequence of necrotizing fasciitis. Genomic analysis revealed that the virulence genes were transferred between the bacteria, indicating a significant role of horizontal gene transfer in the infection. Microbiology connection, a beneficial mutation. Since the first reported case of human immunodeficiency virus in 1981, nearly 40 million people have died from HIV/AIDS. The virus specifically targets helper T cells or CD4 T cells, which are crucial for the immune system's response to infections. While there is currently no cure for HIV, various medications have been developed to slow the virus's progression. The highest prevalence of HIV infection is among individuals ages 15 to 49 in sub-Saharan Africa, where approximately 1 in 20 people is infected, representing over 70% of global infections. Unfortunately, access to prevention strategies and treatment options is severely limited in this region. Recent scientific discoveries have highlighted a few individuals from Northern Europe who possesses a resistance to HIV infection. In 1998, a genetic analysis led by American geneticist Stephen J. O'Brien revealed that a significant percentage of individuals of Eurasian descent carry a deletion mutation known as CCR5 delta 32. This mutation occurs in the gene for CCR5, the co-receptor for the T cells essential for many HIV strains to penetrate the host cells. Those homozygous for this mutation show significantly reduced susceptibility to HIV, while heterozygous individuals possess some level of protection. The reasons for the prevalence of these mutations among Northern Europeans remain unclear, although its frequency decreases as one moves southward. Research suggests that the mutation predates the emergence of HIV and may have been favored by historical exposure to the disease like plague or smallpox. The mutations seems to confer protection against these diseases, which could explain its higher occurrence in the region that faced such epidemics. Current estimates place the age of this mutation between 175 to 225 years ago, with the possibility that it spread through Viking invasions. These findings have opened new research paths in HIV treatment, notably the exploration of drugs that block HIV's binding to CCR5 in individuals without the mutation. While DNA testing for the CCR5 delta 32 mutation is technically feasible, instances of individuals with the mutation still contracting HIV exist, leading to public health officials to discourage widespread testing. This is to avoid promoting risky behaviors among those who might mistakenly believe that they are entirely immune. Nonetheless, targeting CCR5 remains a promising strategy in developing effective therapies for individuals living with HIV. Now, there is an FDA-approved drug Maraviroc that is available to the patients, which actually block the CCR5 co-receptor and prevents the virus from entering the cells. Clinical consequences of transduction. In 2011, Paul, a 23-year-old relief worker from Atlanta, traveled to Haiti to assist in recovery efforts after the 2010 earthquake. After several weeks of work, he began experiencing severe abdominal distress, including intense cramping, nausea, vomiting, and watery diarrhea. He also suffered from significant muscle cramping. At a local clinic, the physician suspected cholera due to an outbreak following the earthquake. Cholera spreads through the fecal-oral route, and the disruption in sanitation, which often occurs after natural disasters, can lead to outbreaks. The physician confirmed the diagnosis using cholera dipstick test and prescribed Paul a single dose of doxycycline along with oral rehydration salts, advising him to consume plenty of clean water. Its symptoms primarily arise from the cholera toxin, which activates the chloride transporter, causing the chloride ions to be transported from the epithelial cells into the gut lumen. To neutralize the excessive negative chloride ions in the gut, positively charged sodium ions are pulled into the gut, which forms sodium chloride. Salt attracts water, and this process leads to excessive water loss into the gut, resulting in a severe watery diarrhea associated with cholera. Environmental Vibrio cholerae are found in water bodies and can harbor genomic islands by horizontal gene transfer, rendering it pathogenic. Pathogenesis of toxigenic, toxin-producing Vibrio cholerae critically depends on the production of cholera toxin, which is responsible for cholera symptoms, and the toxin co-regulated pilus, or TCP. The genes for the cholera toxin, CTX, are from the filamentous bacteriophage CTX5. That has been incorporated into the genome. The genes in the TCP island encode factors necessary for the colonization of the small intestine in the human host after ingestion of contaminated water. Therefore, pathogenic strains of Vibrio cholerae arise from the horizontal gene transfer via specialized transduction. Here are some links to learning and educators' resources. Thank you.